Latex injection makes visible a single nephron in this rat kidney at a magnification of ×60. It's the sum total of the functioning of about 30,000 nephrons that carries out the excretory and osmoregulatory functions of this kidney. Let's remind ourselves of the structure of the kidney. These paired organs are found towards the dorsal side of the abdominal cavity in mammals. In this model, we can see that there are two distinct regions to the kidney, the outer cortex and an inner medulla. Blood enters the kidney via this renal artery and leaves via the renal vein. In between, blood is filtered within the kidney, forming urine, which passes out of the kidney via the ureter, which is here. In this preparation, the veins have been coloured blue and the arteries red. The renal artery divides, eventually forming the afferent arterioles, which supply individual nephrons. Each nephron begins with a structure called the glomerulus, where blood is filtered to produce the primary urine or glomerular filtrate. Looking at the blood supply to a glomerulus by scanning electron microscopy, we see that the afferent arteriole, which supplies the blood to the nephron, is much wider than the efferent arteriole, which takes it away. This results in an increased resistance to blood flow in the efferent arteriole and a relatively high pressure within the glomerulus. This pressure forces an ultrafiltrate of plasma out of the capillaries, and this primary urine passes along the nephron. Some of the tissue has been removed from this glomerulus, showing the red blood cells within the capillaries. Fluid passes along the proximal tubule to the loop of Henle, to the distal tubule, and then to the collecting duct. And the collecting ducts drain into the renal pelvis and then onto the ureter. Now, the composition of the glomerular filtrate has changed greatly from the glomerulus to the final urine. Usually, it's much more concentrated. The photomicrographs of the sort we've been looking at can tell us a great deal about the structure of the nephron, but they don't tell us very much about how it functions. For this, we need specialized in vivo techniques. We're going to look at some of the work currently being carried out in the physiology department at the University of Manchester. Dr. Hugh Garland.
The experiments that we run in Manchester are almost exclusively confined to the anaesthetized rat, and the preparation that we use looks like this. The first thing that we do is to place two catheters into one of the jugular veins. The first of these allows us to infuse an isotonic saline solution, and the second is used to inject drugs and hormones without interrupting the continuous infusion. We also place a catheter in one of the carotid arteries in order to measure blood pressure and to ensure that it stays constant. Moving on to the kidney itself, we've separated the left kidney from any surrounding fat and connective tissue and exposed the surface, and we're holding the kidney in a small cup. The reason for this is to prevent any vibration or movement of the animal, such as respiratory movement, from being transmitted to the kidney surface. There's a small opening in one side of the cup through which the ureter passes, which would normally convey the urine to the bladder, but as you can see, we've cut the ureter and put a catheter in so that we can collect urine directly. We can use such a preparation to carry out a variety of experiments. One of the first things that we do is to inject a small amount of a dye called lissamine green into the jugular vein. If we look at the surface of the kidney as this is done, we see an immediate effect. This is the injection of the lissamine green, and within a couple of seconds the entire surface of the kidney becomes green. But by about 12 seconds or so, the colour is beginning to fade away. But to understand what is happening, we need to look more closely at the structure of an individual nephron. The tubules that we're looking at are superficial cortical nephrons, which have proximal and distal segments that lie very close to the surface of the kidney. The lissamine green reaches the kidney very rapidly and is filtered at the glomerulus and passes on into the proximal tubules, and these become green. But the loop of Henle, of course, lies much deeper in the kidney, and as the dye moves down and around the loop, the surface of the kidney resumes its normal colour. At around 30 seconds or so, the dye reaches the distal tubule, so parts of the kidney surface become green again. Then, as the tubular fluid drains on into the collecting tubules, the kidney surface reverts to its normal colour, and the dye begins to appear in the animal's urine. Let's have another look at the actual experiment. Here we see the kidney surface again. Notice that before the injection, the urine is completely clear. The dye reaches the kidney surface very rapidly and can be clearly seen within the proximal tubules. At around 11 or 12 seconds later, the surface begins to clear as the dye moves on into the loop of Henle. The time taken for the dye to pass through the nephron is always measured at the beginning of an experiment in order to check that the kidney is functioning normally. At around 29 or 30 seconds, the dye reappears in the superficial distal tubules. It's because the dye is moving around all of the nephrons synchronously that the whole kidney changes colour so abruptly. This technique indicates that the kidney as a whole is functioning normally. The colour is now beginning to fade again as the dye in the tubular fluid passes on into the collecting tubules. We can see that the dye is also starting to show in the final urine, which now appears green. By about 2 minutes, most of the dye has been cleared from the kidney, which has returned to its normal colour. So this technique indicates that the kidney is functioning normally. But it also tells us which are the proximal and which are the distal tubules, because the proximal tubules become green before the distals. So having established which are the proximals and which are the distal tubules, we can begin to investigate exactly what's happening within a specific nephron segment using a technique called renal micropuncture. In the rat, the proximal tubule is around 30 microns in diameter, and it's possible, using a micromanipulator, to insert a very fine micropipette into a tubule so that we can withdraw or inject fluid into a single nephron. One technique is the so-called split drop method, which we can use to quantitatively measure fluid absorption within the proximal tubule. We're using a double-barreled micropipette with black oil in one barrel and a Ringer solution in the other to puncture a straight segment of a superficial proximal tubule. And the first thing that we do is to fill the tubule with oil. The oil is then split with a small column of Ringer solution injected from the second barrel of the same pipette. Now, since the proximal segment has the ability to reabsorb salt and water, the Ringer solution will be absorbed into the surrounding capillaries, and the two oil menisci begin to approach each other. In order to measure the rate of fluid absorption within the tubule, the process is filmed and individual frames subsequently analysed. This used to involve the film being projected and measurements made to calculate the changing volume of the droplet. But this was found to be extremely subjective, and so we now do the analysis using more sophisticated methods. We're using an image analysis system to look at individual frames that we've recorded on film down a microscope. And we have the first frame in such a sequence on screen. I'm using a light pen to indicate approximately the position of the two menisci. The computer is now accurately redrawing the lines that I've just drawn, and it's going to measure the distance between the two menisci and also calculate the diameter of the tubule concerned. We've now moved on to frame 10, and you can see that the droplet has shrunk and the two menisci have begun to approach each other. And we can get the computer to automatically draw in the two new menisci. Normally, of course, we would analyze a whole series of frames in any one sequence. When we do this, we find typically that the rate of fluid reabsorption in the proximal tubule is around 2 nanoliters per millimeter of tubule per minute. But this technique, of course, will only measure the rate of absorption of a fluid that we've introduced into the tubule. We can find out much more about what's happening in a functioning nephron using a technique called free-flow micropuncture. For this technique, a single-barreled micropipette is inserted into a proximal tubule, and we inject a bolus of oil. We then withdraw tubular fluid, holding the bolus steady, and you can see fluid beginning to collect in the pipette on the right. If we withdraw tubular fluid at exactly the same rate that it's reaching the pipette, the oil block shouldn't move either upstream or downstream. Over about 5 minutes, a small amount of fluid will be collected, typically around 80 nanoliters or so. And we can use this volume to analyze for ionic concentrations such as sodium, potassium and chloride, and also for total osmolarity. But the amount of fluid that we collect does not tell us how much is being filtered at the glomerulus in the first place, because some fluid will have been reabsorbed earlier on in the tubule. In order to account for this, we can infuse the compound inulin into the animal. This compound is freely filtered at the glomerulus, but subsequently remains unaltered in the nephron. In other words, it's neither secreted nor reabsorbed. So any increase in the concentration of inulin within the tubule must reflect the reabsorption of water from the tubular fluid. This graph presents micropuncture data which show inulin concentrations expressed as tubular fluid to plasma ratios at various puncture sites along the proximal tubule. The further along the proximal tubule we go, the higher the ratio. This indicates that a progressive fluid reabsorption is taking place. We can analyze fluid in the distal tubule in the same way. Looking now at the nephron as a whole, we'll just consider 4 sample sites: the glomerulus, the late proximal tubule, the early distal tubule, and the end of the collecting tubule, in other words, the final urine. The amount of fluid reabsorption at each point is calculated from the concentration of inulin. In the glomerulus, there has been no reabsorption. But by the end of the proximal tubule, 50% of the water has been reabsorbed, and by the early distal, 80%. However, when we look at the final urine, over 99% has been reabsorbed. The amount of fluid initially filtered by the glomeruli is very large, maybe 100 times the volume of the animal's blood in a single day. But the amount of fluid lost in the urine is remarkably small. If we look at the sodium concentration at the same points along the nephron, the figures look less startling. They rise from 150 millimoles per litre in the plasma to 200 millimoles per litre in the final urine. But this graph is deceptive. For example, if we look at the figures for plasma and for late proximal tubule, the concentration of sodium is the same in both cases, in other words 150 millimoles per litre. But remember that by the late proximal tubule, 50% of the water has been reabsorbed. For the sodium concentration to be the same, we must therefore have reabsorbed 50% of the sodium. So although its concentration hasn't changed, the absolute amount of sodium has decreased by half. This is achieved by actively transporting sodium ions out of the proximal tubule. Water then follows passively, thus keeping the tubular concentration the same. If we look at the rest of the sodium graph, we see that the final urine contains very little sodium, and thus the amount of sodium lost in the urine is also very small. If we inject latex at the point where tubular fluid has been withdrawn, we can relate ionic concentrations and water reabsorption to a precise point along the nephron. Once the cast is set, the tubule can be dissected out and measured, and the sample point recorded as its exact position along the nephron. But although we can analyse fluid composition in superficial proximal and distal tubules, we can't sample fluid from the loop of Henle because this lies far too deep within the kidney. We can use micropuncture techniques, however, to actually perfuse the loop of Henle. But these will have to be short loops because only these are directly accessible from the surface of the kidney. In order to perfuse the loop, a pipette is inserted into the proximal tubule and a small amount of Lissamine green is injected. As this flows through the nephron, it identifies the position of the last proximal convolution before disappearing down the loop, and the first distal convolution is indicated by the point at which it reappears. A second pipette is then inserted into the last proximal convolution. This pipette is attached to a microinfusion pump, and the loop is perfused with a test solution. The first pipette is now replaced with one containing oil, and an oil block is injected. This ensures that only the test solution goes around the loop. This pipette is then removed and subsequently used to inject a distal oil block, which prevents any test solution moving along the distal tubule. The pipette is then used to collect the perfusate.
So the short loop of Henle can be isolated in vivo, and by analysing the composition of fluid before it enters the loop and when it leaves the loop, it's possible to investigate the role of the loop in handling water and solutes. But remember that the long loops of Henle are not directly accessible from the surface of the kidney, and these loops are very important in urine concentration. Also, the function of the parts of the loop of Henle vary along its length. Some regions function very differently from others. So how can we investigate these variations? The loop of Henle can be isolated in vitro. The loop is dissected out and short sections are perfused. And if the function of each of these sections is examined in turn, it's possible to build up a complete picture of how the loop of Henle functions. These experiments are normally carried out in the rabbit kidney, mainly because it's much easier to tease out the tubules of the rabbit kidney than of the rat. There are differences between the rat and the rabbit kidney, but the general principles of urine concentration are the same in both species, and you should remember this in the next section, which deals with contracurrent multiplication in the rabbit kidney. In the thick ascending limb, chloride ions are actively pumped out. This produces an electrical gradient, negative outside and positive inside, so sodium ions follow passively down this gradient. In the thin ascending limb, both sodium and chloride ions move out passively down a concentration gradient. Now the ascending limb is almost impermeable to water, but the descending limb is very permeable to water, and because of the increased osmolarity in the medullary tissue between the limbs of the loop, water moves passively out of the descending limb. But the descending limb is not permeable to ions, and a difference of about 200 milliosmoles occurs at any horizontal level between the descending and ascending limbs. As fluid moves around into the ascending limb, this small osmolarity difference is magnified into a large osmolarity gradient from the cortex to the medulla. This process is called contracurrent multiplication. The overall effect of these ionic and water movements is that there is an osmolarity gradient in the kidney, from 300 milliosmoles in the cortex to around 1200 milliosmoles in the medulla. As fluid moves down the collecting duct, the increase in osmolarity in the medulla causes water to move out passively. But the first part of the collecting duct is not permeable to the excretory compound urea, which becomes concentrated. But further down, the collecting duct is permeable to urea, which moves out and contributes to the osmolarity gradient. Urea also moves into the ascending limb and is recirculated. You will remember that between the early distal tubule and the end of the collecting duct, water reabsorption can increase from 80% to over 99%. Water reabsorption is increased by antidiuretic hormone, which alters the permeability of the collecting duct. While over 99% of the water can be reabsorbed, if the animal drinks a lot of water, less antidiuretic hormone will be released. This results in less water reabsorption, and so large quantities of dilute urine will be produced.
This program has looked at some of the specialized techniques available for the study of renal function, and we have used some of these to describe normal kidney function. Much of our current research uses the same techniques to describe kidney function under altered circumstances. We are looking, for example, at the effect of specific drugs and hormones on the kidney, and also how the kidney behaves under altered physiological conditions. During pregnancy, for example, we found that glomerular filtration rate increases, but the tubules partially compensate for this by actually growing in length, so the amount of fluid reabsorption also increases. As yet, the mechanism responsible for these changes has not been fully elucidated, and this is just one of the topics currently being researched.